Hi team, Jack Orty here, and today I'm going to be taking you through the structure and biology of bacteria. So let's just jump straight into it. Oh, what's going on? Here we go. Okay, cool. So, as I mentioned in the previous video, here is an electron microscope image of a bunch of bacteria. And we can see that there's a number of different species and they have a number of different attributes. So in the previous video, we just talked about how you can divide these bacteria into gram-negative and gram-positive bacteria based on how they stain with a gram stain. And this stain corresponds to a structure in the cell wall, whether they have a lot of peptidoglycan or not a lot of peptidoglycan, which is gram-negative. Now we're going to talk a little bit about shape. Now there's loads and loads of shapes. There's way too many to go through. And it's, unless you really get into this field, it's a little bit of a waste of time learning all these Latin names for the different shapes of the different bacteria. But I'm going to take you through a few so we can understand what's going on with some of the more famous bacteria. So the first one is Bacillus, and the plural is Bacilli, and that's because it's Latin. We're going to see this lus to li transition all the way through this video, so Bacillus to Bacilli, and this is most famously seen in E. coli, and basically it's a rounded rod. It looks like a rod or a rounded square. That is a bacillus shape, and E. coli is the most famous for it. Now, E. coli live in your intestine. They're a very common research bacteria. We use them all the time in the lab. But also, they can cause disease when you get sort of dysbiosis of the gut and you eat too much E. coli. So they're an opportunistic pathogen that can cause disease, even though they often just live harmlessly in our body. And actually, they produce vitamin K for us. So they can do some really nice things in our intestine too. Next, we have Coccus, and the plural is cocci, and that's just a ball. It's just nice and easy. That's a sphere right there. Next, we have Staphylococcus, and the plural is Staphylococci. And an example is Staphylococcus aureus, which is a very common opportunistic pathogen. And so it can cause skin lesions, and it can also cause lung problems by causing infections in those sites. Now, it's often described as a bunch of spheres, and it's often described as a bunch of grapes, and which is quite appropriate because it is a gram-positive bacteria, and that's why it's purple, and so it does look like a bunch of grapes. But not all cocci are gram-positive, and not all bacillus are gram-negative, but E. coli is gram-negative and Staphylococcus is gram-positive. So there isn't a correlation between shape and whether or not you're gram-positive or gram-negative. I've just used the appropriate color given the example bacteria I'm using. So Staphylococcus is gram-positive, so I am showing it in purple. Next we have Streptococcus, which is a long chain of spheres like this. And this is an example bacteria right here, and we've all heard of strep throat. So it's again an opportunistic pathogen, and it can cause diseases in our throat epithelia, for example. And these are famous bacteria. These are used in research a lot. They cause a lot of diseases, and so that's why you've probably already heard of all of these kinds of bacteria, and that's why I'm teaching you their shapes. Now let's do a bit more of a deeper dive into some of the other structures of the bacteria. I've taken you through some basics, but let's do a little bit more and look at some of the more eccentric properties of bacteria. So this is an E. coli bacteria here. Here we have this long protrusion coming from it, and it's actually quite thick, and this is the flagellum, and the plural is flagella, just to keep it confusing. Now, the flagellum is actually a motorized protein that comes out of the bacteria. Now, some bacteria have a flagellum just at one end, and that allows it to get directional locomotion, right? So this flagellum is a powered corkscrew-like protein that can help propel it through through fluid. Now, E. coli is kind of interesting. It's got flagella in every direction, so it just kind of helps it jiggle and move at random, and it helps it go through fluids, but sort of has that less directional role and more just about trying to get afloat and moving around the fluid medium. Now, this is what the protein complexes look like. So there is essentially a motor. Look at that thing. It is a molecular motor. It's just It's absolutely amazing, this protein structure, and it's embedded in the cell wall, and out comes a passive protein tail. So that tail is passive, but it's all powered by this motor, and you can see that corkscrew-like action here in this computer animation, and that helps it push its way through the fluid, a bit like an Archimedes screw, which is one of the first sort of propeller designs. And so that can help move it through the fluid. And that's really what flagella are mostly about. They're mostly about locomotion. But we actually have more processes coming out of bacteria. This is a coli again, and this is a Fimbriae. And now this is a finer and shorter process than the flagella, and it is not motorized. That's the important thing. It passively sticks out of the cell wall. Now, on these proteins that come out of the bacteria, these proteins are designed to help with adhesion. And so they can help the bacteria adhere to, say, your intestinal epithelial cells, so it can start building a colony and gain a successful infection. Now, because it helps with infection, we have a special term for it, and that's a virulence factor. If there is something about a pathogen that can help it infect you, it's called a virulence factor. And that's really important because, for example, we sometimes mutate E. coli to remove these Fimbriae. And if we take the gene for the Fimbriae out of the E. coli, we really drop its virulence. It can't actually stick to any of our cells, and so it stops becoming a pathogen. It cannot infect us and cause disease. And that's really handy if you want to go on and research it, for example. And that's because we've removed a virulence factor, and a virulence factor helps it become a disease. But next, and if this doesn't excite you, if this doesn't rock your socks off, I think you should give up on studying biology, because this is one of the most insane things I know of in biology, and it's super cool. This is called a pilus, and the plural is a pili, and it's another process that can stick out of bacteria. Here we have two E. coli, and you might guess what it does based on the fact that it sticks these two bacteria together. Let's have a look. So under a transmission electron microscope, which is a kind of microscope where you can look through tissues, we can see that the pilus is actually hollow, which is pretty crazy, right? It's pretty crazy. So the pilus is actually hollow, and what it can do is it can help fuse two bacteria together, and then it can widen, and now we can end up with a process called horizontal gene transfer. So as I've covered in a previous video, bacteria have their main chromosomal DNA, which is a giant loop of DNA that contains all the necessary genes for life. But then they also have smaller loops of DNA called plasmids. And these plasmids can code different genes that they may or may not need. And in horizontal gene transfer, you can actually share the plasmids. And it goes something like this. The pilus connects these two E. coli together, and then one bacteria might duplicate a plasmid. And then it might send that plasmid down the pilus into the other bacteria. Now, this is the closest approximation to bacteria having sex. They have now shared their DNA, and so sometimes it's called a pilus sex organ, and so a sex pilus. And so it's a way of sharing these plasmids. Now, why would you want to share these plasmids? Well, perhaps this plasmid codes for an exceptionally adherent Fimbriae that specifically allows that bacteria to target that mammal. And then it would want to share that DNA, allowing the other E. coli in the colony to invade this. Or perhaps it encodes for a gene that gives it antibacterial resistance, allowing them to share that gene. Now, as I mentioned in a previous video, one of the key things for evolution is variation. So by sharing these plasmids around, they can actually create different combinations of genes in different bacteria, and that creates greater diversity, greater variation in that population, so they can evolve and survive selection pressures, which is absolutely amazing. It's such a cool idea that your bacteria are basically having sex. It's not technically sex. There's no, it's not technically sexual reproduction, but it's very similar, and it's basically the bacteria having sex in your gut all the time. Something worth thinking about. So let's try to tie this all together. Here we have an E. coli. It's got a cell wall and a cell membrane, remember? And if it was gram-negative, it would look like this. It would have two cell membranes with the cell wall, a peptidoglycan layer in between. And the peptidoglycan layer is rigid, which is what allows bacteria to generate different shapes. The cell membrane is not rigid enough, so they would just form round sac-like structures without that rigid peptidoglycan layer. Gram-positive has a very large peptidoglycan layer there, and then just a single phospholipid bilayer. And because this is a coli, it's going to be gram-negative in this example, because they have gram-negative bacteria. Next up, we have that circular chromosomal DNA, and that gets translated into RNA, and then that goes on to ribosomes, and the ribosomes turn that into protein in a process of transcription and translation. And it's kind of similar to what happens in us, but it's not quite, because in eukaryote cells, the RNA undergoes processing and it's much more complex because there is a nuclear membrane. It's way, way more complicated in a eukaryote cell. In a bacteria, it's quite simple. And in fact, the DNA can be being transcribed into RNA and translated into a protein at the same time because there's no nuclear membrane. So it can all happen right there, which is quite convenient for the bacteria. And then we have the last little bit, which is plasmids, small loops of DNA that contain not necessary, but useful, potentially useful genes, and it can help with variation. And lastly, we've got the things that stick out of it. We've got flagella for motility. They're the biggest. They're motorized. We've got pili. These are the second biggest. They're hollow, and they help for horizontal gene transfer. And then we have Fimbriae, and these are virulence factors that help the bacteria stick to the surface of a host cell if they want to invade and become an infection in that host. So we've covered sort of the structure and some of the basic structures of bacteria and what they do, but how do bacteria actually cause disease, right? It doesn't seem like they're particularly dangerous right now. So in the next video, I'm going to go over how bacteria actually lead to disease.